Hello, and thank you for watching this presentation. My name is Dr. Heather Allaway, and I am an assistant professor in the School of Kinesiology at Louisiana State University. And I'm going to talk to you briefly about female astronaut health and the use of animal models to understand and advance our knowledge. Menses is the external sign of reproductive cyclicity in human females, and from menarche, or the first menses, to menopause, the last menses, the occurrence of menstrual cycles at regular intervals of 21 to 36 days is very important for many physiologic functions, including maintaining cardiovascular and musculoskeletal health, immune function, cognitive function, reducing the risk of ovarian cancer, breast cancer, and osteoporosis development. My research focuses on a critical gap in our current women's health research, which indicates the need for a more comprehensive understanding of musculoskeletal health in women throughout their reproductive lives. This is due to the growing public health burden of osteoporosis. Thus, it is vital for us to expand our understanding of the impact of modifiable factors on reproductive health throughout adolescence and young adulthood, and the downstream influence on the development of osteoporosis and fracture risk. Not only is this important to individuals on Earth, but skeletal health will be critical to mission success on the Moon and Mars. With half of the Artemis eligible astronauts in the United States Astronaut Corps being female, a further understanding of specifics around women's health and adaptations to the space environment is becoming ever more important. My research focuses in particular on how alterations in reproductive function impact musculoskeletal health. So what do we know? The estrogen exposure associated with normal menstrual cycles in conjunction with regular physical activity are important in women's bone health. Premenopausal women, including female astronauts, suppress their menstrual cycles for multiple reasons. The two most common hormonal contraceptive options used are the combined oral contraceptive pill and the implantable long-acting reversible contraceptive options. LARC options have only a progestin component and come in an IUD or intrauterine device or underarm subdermal implant option. There is some available data to suggest that the beneficial impact of physical activity on bone may be lost when women use an oral contraceptive pill. It is widely known that the injectable progestin-only contraceptive causes bone loss on Earth due to its complete suppression of estrogen production. This same impact has not been observed for the intrauterine or subdermal implant options. For long-duration missions, female astronauts are likely to choose an implantable LARC option due to the menstrual suppressive capabilities of the options, reduced user compliance, as well as reduced risk of radiation damage to the medication in storage prior to use. Currently, we risk sending female crew members off on long-duration missions using a common medication without a clear understanding of the impact on their future fracture risk. There are many ways to assess alterations in different aspects of physiology using ground-based models, as well as having the opportunity to fly your research on the space station. Using ground-based models like hind limb unloading or 6-degree head down tilt mimic microgravity, we can also use different degrees of partial unloading or head up tilt for mimicking Moon and Mars gravity. We often use animals models for mimicking galactic cosmic radiation and understanding the impact of radiation exposure on all aspects of physiology and behavior. We also have analog missions like Tahara, the NEK, Antarctica, the High Seas Base, or Mars Desert Research Station for examining team dynamics, isolation, and hostile environments. However, none of these ground-based analog missions match the opportunity to conduct your research on the International Space Station, whether you work with astronauts or you work with animals. Certain aspects of research that we are just unable to complete in humans for multiple reasons. Though not a perfect model for humans, rodent research provides huge insights into the mechanisms of action in a full physiologic context that we cannot always get to with human subjects in a speedy timeline. Due to the differences in lifespan and accessibility, We are able to garner a lot of information quickly when we design studies using rodents and we maximize the tissues collected and analyzed. Conducting skeletal research in humans and women's health research in particular in space takes quite a long time. And with a postdoctoral position, beginning a new project with astronauts or a bed rest study was just not feasible. So I turned to the experts and learned about the hind limb unloading model, as well as research with rodents. The grant I wrote and sent into the Translational Research Institute for Space Health was successfully funded from a postdoctoral fellowship. It had the aim to investigate the combined impact of an implanted progestin hormonal contraceptive and disuse on bone health. We first had to conduct a pilot dose response study using ultrasound and estrus staging to determine the appropriate dose of progestin for the rodents. The dose we were looking for was one that would mimic the reproductive suppression observed in the majority of women who use implantable progestin hormonal contraception. We hypothesized that the use of the LARC implant would prevent the loss of bone that we normally observe with hind limb unloading. In our unloading study, we had 16 to 18-week-old female sprague dolly rats arrive at our facility. These animals' long bone growth is complete and they are reproductively mature. These animals acclimated at our facility for 3 weeks prior to being randomized to have a placebo or LARC pellet implanted under the skin between their shoulder blades. A week after the pellets were inserted, the animals were randomized again to either weight-bearing or hind limb unloading groups. This resulted in 4 experimental groups which have the following color scheme on all graphs. The placebo weight-bearing animals are in solid black. Placebo hind limb unloading animals will be in open black. The LARC weight-bearing animals are in solid dark red, and the LARC unloaded animals will be in open dark red. Though the hind limb unloading was confusing to the animals at first, they do acclimate to the treatment within the first week and relax. In this study, we maintained our animals in hind limb unloading for 6 weeks, after which the animals were euthanized and the tissues were collected. We collected blood samples from our animals immediately prior to the pellet implantation, as well as right before unloading initiation, and then again at tissue collection. We assessed the tibia bone using peripheral quantitative computed tomography at the start and at the end of the hind limb unloading period. We also injected calcium twice in the final 9 days of the study to assess active bone mineralization that occurred at the end of our treatment. As an assessment of animal health, we monitored body weight and food consumption on both graphs, on all animals. On the x-axis of both graphs, you can see the study weeks across time. There are 3 arrows, one indicating when pellet was inserted, which is blue. Next to it is when hind limb unloading was initiated. And then lastly, over at point 10 is the tissue collection occurrence. Body weight in grams is represented on the y-axis of the left graph. There was no difference among groups in body weight change until 2 weeks after hind limb unloading was initiated, when weight-bearing animals continued to gain weight and the hind limb unloading animals stopped gaining weight and weighed significantly less than their weight-bearing counterparts. Animal food consumption in grams is displayed on the y-axis of the right graph. When animals arrived at our facility, we changed their food to a phytoestrogen-free diet. Initially, the animals ate a lot of this food and their consumption slowly stabilized by week 3 when we inserted the pellet. LARC implantation did not impact their food consumption. However, initiation of hind limb unloading led to a significant increase in food consumption in the hind limb unloaded animals compared with the weight-bearing animals, beginning at the start of HU. The gold standard confirmation that animals placed in hind limb unloading are actually unloaded is to weigh their soleus muscle, which is seen in the red box. This is a muscle that is a postural stabilization muscle. It is constantly activated on Earth due to gravity. The loss of the soleus mass was observed as expected with hind limb unloading, and it was not altered by LARC use. In the blood samples we collected, we measured a marker of bone formation known as P1NP, shown on the y-axis of the left graph, and a marker of bone resorption, or CTX, shown on the y-axis of the right graph. On the x-axis of both graphs are the 3 time points where we assessed these circulating markers. P1 and P changed over time consistently in all groups, measured with concentrations decreasing from week 3 to week 10. CTX, the marker of bone resorption, also changed over time, but in association with pellet group, your solid black versus solid maroon, or your black bars versus your maroon bars, as well as loading group, which are your solid bars compared to your open bars, but independent of each other. Both placebo groups, so black, and LARC groups, maroon, had an increase in CTX concentrations from week 3 to week 4, and a decrease from week 4 to week 10. What was different in this pattern was that the placebo group CTX concentrations at week 10 were lower than those measured at week 3, while in the LARC group, the week 10 concentrations were not different than their week 3 concentrations. At weeks 3 and 4, there was no difference in the CTX concentration between weight-bearing or filled bars and hind-limb unloaded open-barred animals. While at week 10, the CTX concentration was greater in hind-limb unloaded animals compared to their weight-bearing counterparts. For the PQCT assessments of bone health, we're going to start at the tibia metaphysis, which is near the knee. Change in volumetric bone mineral density is shown on your y-axis here, and on the x-axis are the compartments where we assessed volumetric bone mineral density. Change in cortical volumetric bone mineral density, which is shown in the red area on the graphic to the left, was not impacted by either pellet type or loading group, while change in cancellus, or that yellow space shown on the left, was independently impacted by both pellet type and loading group. The LARC implanted animals lost more bone density in the 6 weeks than the placebo animals, which was independent of the large loss in bone density in hind-limb unloading animals over the 6-week period. Overall, there was a greater loss of total metaphyseal volumetric bone mineral density in the hind-limb unloading animals compared with the weight-bearing animals. We then assessed the change in cortical shell thickness at the metaphysis, which is shown here on the y-axis. The value we get is an average of the thickness around the shell, indicated by the black lines interspersed around the bone cartoon. Hind-limb unloading caused a significant thinning of the cortical shell over the 6-week period. We also assessed cortical volumetric bone mineral density at the mid-shaft of the tibia. And it was impacted by pellet type, where there was a greater increase in volumetric bone mineral density in the LARC animals compared with the placebo animals at the mid-shaft of the tibia. Differently, there was a change in cortical thickness that was distinctly different based on loading group, where there was not an increase observed in cortical thickness in the hind-limb unloading animals, while there was a large increase observed in the weight-bearing animals. We used the calcine assessment or injections to assess mineralization at the metaphysis, shown in the black bar, which is where the PQCT was measured, as well as at the diaphysis, or the mid-shaft of the tibia, which is where our PQCT was also measured, that we just talked about. At the mid-shaft of the tibia, we assessed the periosteal and the endocortical surfaces for this green label. And in the metaphysis, we assessed all bone that was not cortical bone. What we were looking for was differences in the amount of single label and double label to determine bone formation rate. What we observed is that disuse reduced the overall bone formation rate at the diaphysis on the periosteal surface, but it did not have a significant impact on bone formation on the endocortical or inner surface of the diaphysis, or on bone formation on the cancellus bone in the metaphysis. So the reduced weight and increased food consumption observed in these animals indicate that there is either an absorption or a utilization of energy disruption. Our resorption and formation data from bone indicate, or from the blood indicate, that our animals are experiencing similar things to the astronauts, where there is an increase in bone resorption during flight with no change or a decrease in bone formation. The LARC implantation and hind limb unloading had different impacts on the metaphysis and the diaphysis of the tibia. Unloading decreased volumetric bone mineral density at the metaphysis, which occurred early in unloading based on the absence of a difference in mineralization during the final week of the intervention. LARC use increased volumetric bone mineral density at the diaphysis, while unloading reduced cortical thickness and active mineralization on the outer surface of the cortical bone of the tibia. On balance, the LARC implants did not change the impact hind limb unloading had on the unloaded bone. It did not prevent the loss of bone in the hind limb as we hypothesized, but it did not make it worse, which is key, leading us to suggest that LARC is a viable choice for menstrual suppression in premenopausal astronauts for their exploration class missions. The research I have conducted to date is not done in isolation. It was done as a result of collaboration within the labs that I have been a member of across multiple universities in Canada and the United States, as well as with the labs that I was fortunate to collaborate with outside of my primary lab. I am forever grateful to the research participants who spent time on every project, as well as I am grateful for the lives of the animals that were sacrificed to enable me to gain a better understanding of reproductive and musculoskeletal health. None of the projects I have been involved in could have been completed without multiple funding sources, and specifically my postdoctoral funding through the Translational Research Institute for Space Health. Thank you for taking the time to watch this video, and hopefully you will join any multitude of other sessions. Goodbye.